Sorry, I got cut off in the middle. Over here on the lower right, what you can see is a very, very magnified view showing the centromere, and you can actually see the kinetochore microtubules and how they are attached to the kinetochores. I love these images. And again, there's never too many images of this. For some reason, it's one of my favorite things. Oh, there they are again. Okay, now next phase is metaphase, and meta means middle, of course. And so what happens at this point is that you have those chromosomes lined up in the middle or at the equator. So centrosomes, as you can see, are at opposite poles, and the chromosomes are at what we call the metaphase plate. And there's no actual structure, it's just the middle of it. And then centromeres are lined up in the middle. There are approximately 15 to 35 kinetochore microtubules that are attached to each kinetochore of the centromere. So imagine the number of microtubules that are there. Then you have all these other non-kinetochore microtubules that are coming out of the centrosome at each pole. Now, anaphase is following metaphase. And in anaphase, the actual phase begins when centromeres move apart. So in this image over here, you can see that these sister chromatids have now separated, and each is their own chromosome. Now, if you realize, of course, too, that when DNA was replicated, it produced an identical chromosome, then you can see here that those sister chromatids, which were identical, are now splitting and forming into two new cells. And so each of them has its own centromere, so each of them is a sister chromatid. They are pulled to opposite poles, and again, they're pulled centromere first. You can see that right there. And then the way that those chromosomes are pulled, and this is one theory, is that what will happen is the kinetochore microtubules, which are the ones that are attached to the chromosomes at the kinetochore, shorten, and that pulls the chromosomes. And then in addition, you will also see that this cell is no longer a sphere, a circular structure. It's becoming more elliptical because the poles are moving apart, and that elongates the cell. And at the end of anaphase, each pole will have its own complete set of chromosomes, and we are on our way to two daughter cells. Now, this is the experiment that was done. What you had was, and this is where this came from, the belief that the microtubules are actually shortening, and that's where that's how they separate. Here's the spindle. Here's the kinetochore. And the experiment is they used markers, and they kept those markers and viewed them to see where they were after a part of anaphase had occurred. And you can see that there's less microtubule material here versus in this image, and that is because the microtubules shorten in order to pull the chromosomes. Now, you remember the term polymerization, that is the forming of a polymer or the forming of a micro or macromolecule. The opposite is depolymerization, and that's what's happening here. And here's another view. They depolymerize at their kinetochore ends, and you can see that parts of the microtubule are shown breaking off here. Last is telophase and cytokinesis. Telophase, when telophase occurs, cells further elongate, and you can see that it's even longer. Daughter nuclei have formed. The nucleoli reappear. Chromosomes form chromatin again, and by the end, the nucleus is divided into two identical nuclei, and we call those daughter cells. There's a structure called the cleavage furrow that forms during cytokinesis. And what happens is there's a contractile ring that reduces the diameter of the cell, and the mitotic spindle disintegrates. And we'll watch this in the next video.